Chapter 24, the immune system. In this chapter, we'll talk about the anatomy and function of the immune system. We'll look at how our body responds to different types of pathogens and what those pathogens may be. We'll discuss innate and acquired immunity and take a look at an example of how we might defend ourselves against bacteria and viruses, and then just end with a discussion on how the nervous system, endocrine system, and the immune system interact. So, what's the purpose of the immune system? Well, it's to protect us. And in order to do that, it has to be very specific about who it's going to attack, so specificity. And we also want our immune system to have the capacity to remember. So, memory is another important aspect. Being able to be specific about who they attack ensures that our body will be able to distinguish self from non self cells. Again, this is all when everything is working normally. Our immune system will ensure that it only recognizes cells that are abnormal or altered or foreign, and that it will be able to understand when it comes across a cell that is one of our own, and so it doesn't mount an immune response. So, that being said, the three functions of the immune system is to protect us from pathogens, foreign molecules that may invade our body. So, parasites like hookworms and tapeworms, bacteria, and viruses. It also removes dead or damaged cells or cells that have become abnormal. So, not only does it protect us from foreign things, it also protects us from our own cells that have become damaged or altered. If things don't work quite right, then pathologies can develop. And here are kind of the main ways or main types of pathologies that are associated with the immune system. The first is an incorrect response. And so these are autoimmune diseases. And we've talked about, for example, type 1 diabetes. So an example of an autoimmune disease is when our own immune cells attack our own body cells. So in type 1 diabetes, our immune cells attack the beta cells of the pancreas. So that would be considered an incorrect response. So in this instance, it's not recognizing or distinguishing self from non-self cells, and as a result, it's attacking our own cells. Another pathology is an overactive response. So in this case, we have a response that's bigger than necessary. So an allergy is a great example of that. So you get a response, and you do need a response to an allergen, but it's out of proportion to the threat. So the response is just much bigger, and as a result, it starts to have negative consequences on our body. And the third is a complete lack of a response. So the example is an immunodeficiency disease like AIDS, where components of the immune system just fail to work. And as a result, as you might suspect, you're going to have increased incidence of illnesses and sicknesses and pathologies develop. So, what are the different types of pathogens that can attack us? Two examples are bacteria and viruses, and you'll discuss more about them when you get into microbiology, but some general differences include its structure. So, bacteria are cells, so they're usually surrounded by a cell wall, whereas viruses are not, so they have a nucleic acid core and just a protein coat. Because bacteria are cells with a cell wall, they can survive outside of a host. They are also able to reproduce outside of a host. Viruses cannot, so they have to be inside of a host to reproduce. As a result, they are parasitic. How do we kill bacteria? Antibiotics. So when you go to the doctors, a lot of the times, depending on what you have, again, if it's a bacteria, you'll get antibiotics. And we have lots of different types of antibiotics. And that's because our antibiotics can be developed to attack bacteria. And since bacteria live outside of a host and reproduce outside of a host, it's easier to target them. Versus viruses reproduce inside of a host, so it can be a little bit trickier. And one thing that can happen is you can get something called host toxicity if the drug isn't working quite right. So it's a little bit more challenging. But we do have antiviral drugs to target viruses, just not as many as antibiotics. So this is just an example of a virus. So we have a protein coat and the nucleic acid core. So how does our body protect itself from these pathogens that can invade us? So we have two lines of defense. The first is a physical or chemical barrier. That's our first line of defense. So that would include like our skin, right? So our skin is a physical barrier from the outside world to the inside. And so when we have an open wound, for example, that's one way for pathogens to get into our body. Our epithelial lining, cilia, the acids of our stomach, the mucus, lysosomes, all of these are designed to try to keep pathogens outside from getting into our body. So, for example, in our respiratory tract, we have mucus that's secreted. And with the help of mucus, pathogens might get trapped in the mucus, and then the cilia kind of beats it out so that we can then cough it out and it doesn't actually enter into our body. Our stomach is very acidic. Again, it's one way we can protect ourselves before that pathogen can actually get into our body's internal environment. But if this first line of defense fails, then we're going to need our second line of defense, which are your immune cells. And so our immune system will respond with either innate or acquired immunity. So we'll take a look at two. But first, let's make sure we understand what the immune system is composed of. So we have our lymphatic tissue and then obviously our immune white blood cells. So the lymphatic system, we saw this a figure similar, or we've talked about the lymphatic system twice already. So one was when we talked about the blood vessels and how when we filter blood out of our capillaries, anything that we don't reabsorb that gets built up in the interstitial space, the lymphatic system will pick it up and then drain it back into the circulation. The next time we talked about the lymphatic system was when we talked about the digestive system. So when we absorbed fat in our digestive tract, it actually got absorbed into our lymphatic vessels before it got drained into our circulatory system. And here's another function. It's part of our defense, our immune system. So the lymphatic tissue is distributed throughout our body. And we have primary and secondary lymph tissue. So primary lymph tissue is shown here in red. And primary lymph tissue is really just anywhere our immune cells are born or where they mature. So your bone marrow would be considered primary lymph tissue. Your thymus gland, where your T cells mature, would be primary lymph tissue. Everything else is secondary lymph tissue. So your lymph nodes, your spleen, and these are areas where those mature white blood cells are going to interact with pathogens and initiate a response. So here are the other cells that make up our lymph tissue. We have our white blood cells. So our white blood cells, we have several types. We have our basophils and mast cells, our neutrophils, eosinophils, monocytes and macrophages, lymphocytes, plasma cells, and dendritic cells. Let's quickly just go over some of the classifications. So some of our white blood cells or leukocytes are classified as phagocytes. So the neutrophils, the eosinophils, monocytes, and macrophages are phagocytic cells, which means they will phagocytose. So they will actually eat up pathogens. Granulocytes just means that when you look at them, they look like they have little granules in them anatomically. We have some cytotoxic cells like the eosinophils and some of our lymphocytes. And these cytotoxic cells are toxic. So it makes sense that they are going to have a role in destroying. And then we have our antigen presenting cells. Antigen presenting cells, so an antigen is just anything an antibody binds to, and typically antibodies are going to bind to our foreign pathogens that come in. So an antigen presenting cell is an immune cell in this case that will take a piece of that pathogen and present it on its cell surface, hence you are presenting the antigen. We'll come across this and I'll explain it more as we move on. So let's talk about innate immunity. So innate immunity is innate, meaning it's in you and it's always present. It is non-specific about who it attacks. So when the body's first line of defense fails, then we're going to initiate our immune response, and an innate immunity is one of them. It consists of our white blood cells, and they're going to attack and destroy the pathogen. And it's considered non-specific because these white blood cells will pretty much attack any material that they identify as foreign. And so it involves our phagocytes, like our macrophages, neutrophils, natural killer cells. And these are going to phagocytose and destroy the pathogen. And they will attract other immune cells to the area by secreting cytokines. Now, why would you want to do that? Well, that's because when you have one pathogen that a macrophage, let's say, came across, there's probably not just that one. There's probably more. So the macrophage will phagocytose the pathogen that it found, but then it'll release cytokines to attract more white blood cells to the area to then hunt and look around for more pathogens. Also, the inflammatory response is part of innate immunity. So here we're seeing the pathogen, and here's a phagocytic cell macrophage that can phagocytose it. Some pathogens will actually cover themselves with a polysaccharide capsule. In those instances, we might need the help of antibodies. We're going to talk more about antibodies later, but antibodies have several functions, and this is one of them. And its role is to act as opsonins, and that basically means that the antibodies will bind to and tag the pathogen. So what we've done here is we formed an immune complex. So the antibody bound to the pathogen, and now our phagocytic cell is able to recognize that an antibody has found a pathogen. We have an immune complex, and it can phagocytose the pathogen that initially was sort of hiding. So I said inflammatory response is part of innate. So it's a hallmark reaction of innate immunity. And it's created when our macrophages release those cytokines. Remember, the macrophage phagocytoses the pathogen, but to let others know that it found a pathogen and there's probably more in the area, those cytokines will attract more immune cells to the area. So the role of the inflammatory response includes, number one, attracting these other white blood cells to the area, and chemical mediators will also be released. You'll produce a physical barrier to try to prevent the infection from spreading, and then you also promote tissue repair. So basically, you're trying to bring more white blood cells to the area and deal with the infection, promote repair, and obviously there are signs and symptoms associated with inflammation. Other players in the inflammatory response include our complement proteins. These basically create these water pores. So they'll insert themselves into the membrane of our pathogen, creating a little pore, and it's a water pore. So then water will rush into our pathogen, and eventually when enough water rushes in, it will lyse or burst. And these complement proteins, they're also known as these membrane attack complexes. Now we have acquired immunity. Acquired immunity is more specific. So innate immunity was non-specific about who it attacks. Just if it sees something as foreign, it's going to mount a response. In acquired immunity, the pathogen that they're going to go after is more specific. So it's an antigen-specific response. And it involves our lymphocytes. So we have our T lymphocytes. They're also known as our T cells. Our B lymphocytes, also known as our B cells, and then our NK cells, or natural killer cells. So the natural killer cells has the word killer in it, so it makes sense that these are going to attack and destroy, in this case, virus-infected cells or tumors. T lymphocytes, once they're activated, they'll actually develop into helper T cells and cytotoxic T cells. Again, cytotoxic has the word toxic in it, so it makes sense that these guys would also attack and destroy virus-infected cells. Helper T cells have the word help in it, so they are actually going to regulate other immune cells, and essentially they're going to call for more help. B lymphocytes, once they're activated, they will develop into plasma cells and memory cells. Memory cells will stick around and remember. So these are long living and they can reproduce themselves, and they essentially remember the pathogen. Remember that our immune system has specificity and it has memory. This is where the memory comes in. B cells can also turn into plasma cells, and plasma cells are the cells that actually make our antibodies, and antibodies are quite important in the immune system. So let's talk specifically about those B lymphocytes. So the B lymphocytes are involved in what we call humoral immunity. So B lymphocytes will insert antibodies into its cell membrane. Excuse me. And they will, when they're activated, differentiate into plasma cells and memory cells. So plasma cells will make more antibodies, and memory cells will stick around and remember in case we get exposed to the same pathogen. And when we do, we can mount a quicker response. So for example, let's say we were exposed to a pathogen. That pathogen that came in is going to kind of be able to have free reign in our body. So it's not often that as soon as a pathogen comes in, an immune cell comes in contact with it. So that pathogen is going to come in and it's going to start to reproduce, and eventually one of our immune cells, like a macrophage, will find it, it'll phagocytose it, it'll activate our immune system, and eventually we'll start to make antibodies. And if you take a look at the antibody concentration in the plasma, in the blood, it takes about 2 weeks after that initial exposure for our antibody concentration to peak, which means our pathogen has had kind of free reign in our body for that period of time. So we are likely to have gotten sick at this point. Now, why do we want antibodies and why do we look at antibodies? Because when an antibody binds to our pathogen, it creates that immune complex. Well, when that happens, that pathogen can't really go and do bad things anymore. And so we want to flood our body with antibodies, and that will help us from getting sick. Now, when we activated our B cells, not only do we make our plasma cells, which made these antibodies, we also make our memory cells. And those memory cells are going to stick around and remember. So when we have, after that primary immune response that we just talked about, after we have a second exposure to that same pathogen, we are going to mount a secondary immune response. And in that instance, because we have those memory cells will be able to activate our immune response much quicker. And as a result, we're going to make antibodies right away. It doesn't take as long for the process to happen. And so not only are we going to make antibodies right away, we are going to also make a lot more antibodies. And so as a result, during that second exposure, when we activate the secondary immune response, we tend to not get sick because we have flooded our body with so many antibodies that those antibodies are going to bind up those pathogens before they can actually invade our cells and cause damage. And this is what makes vaccinations work. We are essentially creating memory cells. So antibodies are quite important, and we have different classes of antibodies. So another word for antibody is immunoglobulin, and there are 5 classes, and they are Ig. The Ig stands for immunoglobulin. They are IgG, IgA, IgE, M, and D, and I like to remember them using the word gamed, G-A-M-E-D, actually, gamed. So something unique about each of them. So IgG makes up about 75% of the antibodies in adults. So this is the one that's most abundant. These are actually also very small, and as a result, they're able to cross the placenta and give immunity to the fetus and then obviously the baby during the first couple months of life. IgA is found in things that are secreted, like saliva, tears, mucus, breast milk, and so they're able to disable pathogens before they can get actually into our internal environment. IgE, think of allergy. So those are the antibodies involved in an allergic reaction. IgM is part of our primary immune response. So those are the antibodies that actually we're going to make first. And then the antibodies obviously we're going to make the most would be IgGs. So when we have that primary immune response here, or even the secondary, and we have this increase in antibody concentration, we make our IgMs first, but then we make mostly, and then we make our IgGs and we make the most of those. IgD is found on the surface of our B lymphocytes. And then other roles are unclear, but that's good enough. So antibodies are shaped like the letter Y. The arms of the Y have the binding site. So think of these are the hands that will actually touch and recognize and bind. And then the body of the Y is what determines what class of antibody you are, whether you're IgG, A, M, E, or D. So let's take a look at all of the functions of antibodies. So one is they're found on the surface of our B cells. We can insert them into our B cells, and by binding to our pathogen, we can activate our B cells. And again, when we activate our B cells, they turn into plasma cells and memory cells. Plasma cells and memory cells. So memory cells stick around and remember, plasma cells make more antibodies. This one we already talked about. They act as opsonins. So this is when those antibodies will bind to and tag our pathogen so that our phagocytic cell can find them and phagocytose them. They also cause clumping of our pathogens. So because antibodies are shaped like the letter Y, each arm of the Y can bind to a pathogen. And do you see by doing that, we've actually clustered or clumped these pathogens together. And by doing that, it makes it much easier for this phagocytic cell to come and phagocytose them and destroy the pathogens one by one instead of having to go around finding the pathogens because they're all spread out. They also bind to and inactivate the bacterial toxins that can be released by a bacteria. They also activate antibody-dependent cellular activity, for example, in natural killer cells and eosinophils. They can also trigger mast cell degranulation, which is basically when the mast cells falls apart, and when they do that, they release a bunch of chemicals that, again, are involved in the immune response. And then they can also activate the complement. Remember, the complement is when we insert water pores into the membrane, and by doing that, we allow water to rush into the pathogen, and then the pathogen will get destroyed by bursting or through lysis. So lots of different functions. Now let me talk about antigen-presenting cells. So I mentioned them earlier, but basically here we have our macrophage, which is one of our antigen-presenting immune cells, and it has phagocytosed our antigen. Once it eats it up, phagocytoses it, it's going to take pieces of it. So the lysosomal enzyme will destroy that pathogen, break it up, and we're going to take pieces of that pathogen and we are going to present it on our MHC receptors. So these receptors on the surface of our macrophage are called MHC, and specifically these are MHC2. And by presenting it on the MHC receptors, it's able to allow other cells to know that this macrophage phagocytosed a pathogen. So this macrophage is presenting a piece of the pathogen on its cell surface, hence it's an antigen-presenting cell. Now, excuse me, let's talk about our T lymphocytes. So our T cells initiate what we call cell-mediated immunity. And so T cells, when they get activated, we talked about how they turn into helper T cells and cytotoxic T cells. So another thing that plays a role here is that MHC, the major histocompatibility complex. And I'm going to talk about that more here. So there are two classes of MHCs. There's MHC1 and MHC2. MHC2, which we saw earlier, is found on the surface of our antigen-presenting immune cells. So like, for example, our macrophages, our dendritic cells. And what happens is our helper T cells will actually be able to bind to MHC2 receptors. When that MHC2 has an antigen on it, the helper T cells will understand that a pathogen was found and the macrophage is presenting it on its surface, so then it will do what it does, which is call for more help. And one of the ways it does that is to secrete cytokines. Remember, cytokines attract more white blood cells to the area. MHC1 is found in all nucleated cells, so all non-immune, normal nucleated cells. So any cell in your body that has a nucleus, outside of immune cells, have MHC class 1. So the hepatic cells of your liver, the muscle fibers, right? Those cells, the nephrons, all of these cells have MHC1. Scratch the nephron, sorry. What happens when MHC1 has an antigen on it is our cytotoxic T cells will come and bind to it. And it has the word toxic in it. And so these cytotoxic T cells will destroy these cells. So the reason that happens is when a normal cell gets invaded by a pathogen, it's going to take a piece of that pathogen and present it on its MHC1. And so because it's a normal cell that got invaded and infected, we can't repair it. And so the only thing we can do is destroy it. And so that's what the cytotoxic T cells will do. So let's see an example. So our T cells, or T lymphocytes, they are born in the bone marrow, but they'll actually migrate to the thymus gland and mature there. And we have our helper T cells and our cytotoxic T cells. So all normal, non-immune, normal nucleated cells in the body, remember, have MHC1 on its cell surface. If a normal cell got infected, that's the key word, it got infected by a pathogen, it will take pieces of that pathogen and present it on its MHC1. And the reason it does that is because that's how it's going to let other cells know that it got infected. And so cytotoxic T cells are able to recognize when an MHC1 has an antigen on it. And so when it does, it understands that this cell got infected, so it will destroy it. Now, what happens if you don't destroy it? Well, think of it as a virus infected this cell. And didn't we say when a virus infects a cell, it will use the machinery within this cell to make more viruses? So remember, viruses reproduce inside of our own cells. It does that by using our own machinery to make more viruses. And so we don't want that to happen, because if it does, this cell will eventually burst, and now you'll release lots of more virus, a lot more viruses into our body to infect more cells. So to prevent that from happening, the cytotoxic T cells will try to destroy this guy before that happens. Now, on the other hand, instead of our normal cell getting infected, let's say a macrophage found the pathogen and phagocytosed it. So that pathogen went into our macrophage because the macrophage ate it, and now it's going to present it on its MHC2. Because again, when there's one, there's probably more. In this instance, we're not going to destroy our macrophage, because our macrophage didn't get infected. It actually phagocytosed and purposefully brought that pathogen into itself. So instead of just needing to destroy this guy, we're actually going to have our helper T cells come and bind. So helper T cells will come and bind to the MHC2 cells, and notice that there's an antigen. And now it will say, okay, I understand that you found a pathogen. You didn't get infected. You found a pathogen. And so there's probably more around. So let's call for more help by secreting cytokines and activating other immune cells. All right, so let's take a look at how our body would respond to a bacterial infection, and then we'll take a look at a viral. So here we have our first line of defense. For example, our skin, we have a cut, so that's kind of gone. So then the pathogen came in this case bacteria, and now we have to activate our second line of defense, our immune cells. So when the bacteria comes in, there's several ways we can attack the bacteria. The complement proteins can be activated, and they will, again, I mentioned the membrane attack complexes, so they're part of the complement proteins. They will insert themselves into the cell membrane of the bacteria and cause it to lyse or burst. The bacteria coming in can also activate mast cells. So mast cells will actually degranulate. And when they degranulate, it means it falls apart. It actually releases chemicals like histamine. And histamine is an vasodilator. It's also going to increase the permeability of our capillaries. And so white blood cells circulate in our blood vessels. We want more blood to come to this area because it carries the white blood cells. Hence, we will vasodilate. But once the white blood cells get to this area, we need it to be able to leave the blood vessels and get into the site of infection. Hence, we are going to increase the permeability of our blood vessels. So we're actually going to increase the size of our pores so that our white blood cells can get out of the blood into the site of infection. And it will do that because we will also release chemotaxins, which are essentially chemical attractants. So it's like a chemical that attracts white blood cells so the white blood cells know where to go. We also have some circulating antibodies that can bind to the bacteria, remember, act as opsonins and tag them so that our phagocytic cells can phagocytose it. And when our phagocytic cell phagocytoses a pathogen and it comes in, we can activate our B cells, and our B cells can turn into plasma cells, which will make more antibodies so that we can hurry up and tag more of our bacteria. Because again, when antibodies bind to our pathogen, that pathogen can't do bad things because now we have an immune complex. And so the more antibodies we have, then our phagocytic cells can one by one come and phagocytose them. Or our membrane attack complexes can come and cause them to lyse. Now let's take a look at a viral infection. So here we have a virus infecting our cells. So let's say the virus came in and it actually got into one of our normal cells. So it actually infected one of our normal cells. So then our normal cell will take a piece of it and present it on its MHC1. And when it presents it on its MHC1, cytotoxic T cells will come. Because if cytotoxic T cells did not come to destroy this cell, then the virus will use our cell's machinery to make more viruses. And we don't want that. So the only thing we can do is basically sacrifice the cell that got infected. And that's what this cell does. By presenting the antigen on its MHC1, cytotoxic T cells will come and destroy it. So unfortunately, this cell gets destroyed. But the good news is we don't release more viruses. Now let's say on the other hand, the virus got tagged by pre-existing antibodies. So now we have an immune complex, and we like that because when we form this immune complex, that virus can't go and infect more of our normal cells. So it says it cannot infect cells. So that's why we want our viruses. And now these viruses that are tagged, remember these antibodies in this case are acting as opsonins, they will get phagocytosed by macrophages. And the macrophage will then present it on its MHC2 because that's how it lets other cells know that it phagocytosed a pathogen. And when it presents it on its MHC2, helper T cells will come and bind, and it will call for more help. And one of the ways it calls for more help is to secrete cytokines. And here we also see the macrophage itself can secrete cytokines. And the helper T cells will also activate other immune cells, like our B lymphocytes. And when our B lymphocytes get activated, they will turn into plasma cells. And plasma cells make more antibodies. And we like that because these antibodies can hurry up and tag up all of these viruses before they can infect more of our normal cells. So these are examples of how our immune system can respond to a bacterial and a viral infection. Okay, speaking of antibodies and antigens, let's talk about our blood type. So we have an ABO blood typing system. So your blood type is determined by the antigen on your red blood cell. So if you have A antigens, in this case the red circles are A antigens, then you are blood type A. If your red blood cell has B antigens, in this case the blue triangles are B, then you have type B blood. If you have both A and B on your red blood cell, then you're blood type AB. And if your red blood cell has neither A nor B, then you are blood type O. Now remember, your blood is divided into cellular elements and plasma. So the red blood cell is part of your cellular elements. But don't forget, we have plasma, and in the plasma, we have our circulating antibodies. So we'll actually have the opposite antibody in the plasma, because to you, the opposite antigen is foreign. For example, what I mean by that is, if you are blood type A, you are blood type A because your red blood cell it has the A antigen. It does not have any B antigen. So to you, B is foreign. So as a result, in the plasma, you will actually have anti-B antibodies. Or I just call them B antibodies. So these are antibodies that recognize the B antigen. And it's okay to have this in your plasma because you're type A, and so you don't have any B blood in your blood. If any came in, it's foreign. That's why the antibodies are there, because then it can respond. If you're blood type B, you are blood type B because you have the B antigen on the surface of your red blood cell. So to you, A is foreign. And so your plasma will have your A antibodies or your anti-A antibodies. If you are blood type AB, you have both A and B antigens. So to you, neither A nor B are foreign, so you don't have either of those antibodies. Now, you have other antibodies, but you do not have anti-A or anti-B antibodies in your plasma. And then if you're blood type O, you don't have A or O on the surface of your red blood cell. So to you, both of those are foreign. So in your plasma, you will have both anti-A and anti-B antibodies. So why is blood type O considered the universal donor? Now, when you're donating blood, you are donating the red blood cell, okay, not the antibody, the red blood cell. So when you're blood type O, you're the universal donor because on your blood, you don't have any antigens. Okay, you have no A's and no B's. So no matter who you give your blood to, and no matter what antibody they have, there's nothing on your surface to react to. So you can give your blood to everybody. However, you can only receive blood from other people who are O because your blood, while your red blood cell has no antigens, your plasma has both anti-A and anti-B antibodies. So if you were to receive the A blood, your A antibodies would bind to it. If you receive B blood, your B antibodies would bind to the B blood. So the only blood you could receive are other O's because it has no antigens. Now, AB blood is known as the universal recipient. So O is the universal donor, AB is the universal recipient. Why? Because your antibodies in the plasma don't exist. again, I'm only talking about A and B here. You have other antibodies. But if you are AB blood, you don't have A and you don't have B antibodies. So no matter whose blood you get, you don't have any of the antibodies to react to any of the antigens. So you can get blood from everybody. Now, what happens if you mix the wrong blood type? Then what happens is shown over here. You get clumping of your blood. So here we have, the circles are A. Okay, so A blood came in to someone who had O blood. So they were type O. So here's their blood, red blood cell. And because they're type O, they actually have both A and B antibodies. And unfortunately, they got a transfusion of A blood. And so the A antibodies bound to the A blood. And notice, because antibodies are shaped like the letter Y and each arm of the Y can bind, notice it causes clumping of our blood. And we call that agglutination. And think about it. We have blood. It's supposed to circulate easily through our blood vessels. If we have blood that's clumpy, that's not going to happen. Now, I mentioned autoimmune diseases earlier. This is one way the immune system can work incorrectly, where we start to attack our own cells. And these are just examples of the different types of autoimmune diseases our body can suffer from. And this is not an exhaustive list, but we've talked about some of these already. So Graves' disease is a little bit different. Here, the immune system doesn't actually destroy, but it produces our thyroid-stimulating immunoglobulins, antibodies that will actually act like thyroid-stimulating hormone and activate those receptors. Here's our type 1 diabetes, where our immune system destroys our pancreatic beta cells. In MS, multiple sclerosis, our immune system actually destroys the myelin of our central nervous system. In myasthenia gravis, we actually destroy the acetylcholine receptors on the motor end plate. So those would be our nicotinic receptors on the motor end plate of our skeletal muscle fibers, and as a result, your muscles basically don't contract. You lose voluntary control. In Guillain-Barré, we actually destroy the myelin of our peripheral nerves. So you can see how there are actually a lot of different types of autoimmune diseases. And we're going to end with a look at how the nervous system, endocrine system, and the immune system interact. And so basically, it's the idea that there is a link between our mind and our body. And I'm sure you've heard these stories about a couple who've been together for years, and then one dies, and then, pretty soon after, the other one dies as well. Or you've heard the other story where someone was given, a diagnosis that they can only live for 3 weeks, and now they've already they've been able to survive for 3 years already, and they're still going strong. So what's exactly happening? We know that your mind and how you think has a really big impact on our body's function. And the connection isn't completely understood, or what's going on isn't fully understood, but we do know that there's a lot of interaction between the brain, the immune system, the endocrine system. And so how you think and your emotions can influence our body, essentially. And so, as nurses, one of the things that you want to do is not just treat the illness, but really understand that these are people that you are dealing with, and giving them a positive outlook, and not lying to them about treatment, but making sure that they have good perspective. Really improves outcome. For example, therapy dogs have been shown to be really helpful in patients recovering after surgery, for example, because they bring joy and light into people's lives. So, again, just make sure you see the whole person and not just their condition, and looking at them through a chart. So here's one of those stories, and you can find multiple stories like this on, if you do a search online. But this is a Tennessee couple. They died just hours apart after nearly 64 years of marriage. And you can see this really sweet, photo of them in the hospital, just still holding hands. So Dolores Winstead was 83, and her husband, 88-year-old Trent, Trent was admitted to the hospital earlier in the month, and Dolores unexpectedly suffered an aneurysm while she was visiting him, and they ended up passing just hours apart from one another. So Just one of those things to think about. All right, thank you very much.